Okay, so let's take a look at active mechanisms now. So here we're going to use energy to move substances across membranes. All right, and so the first one here is called active transport. So this is a process that's going to move substances across a membrane from areas of low concentrations to areas of higher concentrations. So low concentration to higher concentration. It's going to use a carrier protein. So moving from low to high is the opposite of high to low, obviously. What moves high to low? Well, diffusion moves high to low. All right, so here in active transport, we're going to move against diffusion. We're going to work against diffusion. Molecules want to go high to low. They don't want to go low to high. All right, so in order to get them to go from low to high, moving them across membranes, that's going to require energy. Okay, so a couple examples here. This is showing the sodium-potassium pump. This constantly pumps sodium out of cells, potassium into cells. This is found along all of our muscle cells, all of our nerve cells. If this pump stopped working, you would stop working very quickly. Another one is, an example here is putting iodine into our thyroid gland. Now, if you find your voice box a little bit below there, that right there is your thyroid gland. Now, we pump iodine into our thyroid gland to make a couple of hormones which are collectively known as thyroid hormone. All right, and thyroid hormone regulates our basic metabolic rate. If your metabolism starts going down, you produce more thyroid hormone, your metabolism goes up. Okay, so but we need iodine to do that, and so we constantly pump in iodine into our thyroid gland to make that hormone. And this is why, if you go to the store and you look for salt, and you'll see there on the shelf, it'll say iodized salt. And iodized salt has iodine added to that so that we don't suffer an iodine deficiency. And oh yeah, by the way, about 1 billion people on the planet are iodine deficient. They don't get enough in their diet. If you eat seafood, you're fine, because iodine is found throughout the marine environment, not very common in terrestrial environments. Okay, so moving on here, let's look at membrane-assisted transport. So a membrane-assisted transport, here the membrane, plasma membrane, we're going to use to get stuff into or out of the cell. So the first is exocytosis, and this is showing exocytosis. Exo, exiting, cyto, the cell, right? So there's a transport of substances out of cell by a vesicle. And a vesicle, right here, is a small membranous sac. Okay, so here we have some product here in a vesicle. It's going to fuse with the plasma membrane. It's going to open up and release those contents out of the cell. This is how we get saliva out of our salivary glands, tears out of our lacrimal glands, sweat out of our sweat glands. All right, let's go in the opposite direction, getting stuff into the cell. The first here is, so these are known as endocytosis. Endo means within, so cyto the cell, so within the cell. So this is transport of substances into a cell by a vesicle. So the first is called phagocytosis, and this is trying to show phagocytosis here. So phago means to eat. So this is endocytosis around a large molecule or another cell. So here it's engulfing this other cell, bringing it into that cell. And lysosomes, which we talked about in the lab part here, lysosomes are going to fuse with this phagocytic vesicle, and those digestive enzymes are going to kill that microorganism. So this is how white blood cells are going to kill bacteria. And this is a main defensive mechanism for us, is phagocytosis. So our white blood cells are going to phagocytize other cells that get into us and kill them this way. Next is pinocytosis. Pino means to drink. So, right, so Pinot Noir, Pinot Grigio, those are wines. Pinot Noir is a red wine. Noir means dark. Pinot Grigio, Grigio means light. It's a white wine. Anyway, so this is endocytosis around a liquid, and this occurs in our small intestine, which is our major absorptive organ. So instead of actually going around, now we're going to use negative pressure. We're just going to suck some fluid in, pinch it off into a vesicle. Next is receptor-mediated endocytosis. This is endocytosis around a specific molecule. So here we use a receptor protein. It's going to bind to that particular molecule, and then we're going to draw it into the cell in a vesicle. And this is one of the major ways in which we get cholesterol out of our blood by our liver cells. Cholesterol molecules will bind to these LDL receptors, and we'll draw that cholesterol out of our blood. Two ways I have high cholesterol. One is to eat a bad diet. The other way is to just not make a whole lot of these receptor proteins, and that's due to your genes. Okay. Lastly here is transcytosis. This is a combination of endocytosis and exocytosis used to move substances through a cell. So we can see penocytosis occurring on one side and exocytosis on the other side to move substances through there. Now, if you have my notes, all right, I go into a bunch of structures that are found in our cells. Now, the thing about that is that in the lab portion, we're going to talk about most of these structures. So I'm just going to skip through here and get to things that we have not covered in the lab portion, because there's no need, I feel, to double hit you on these. So looking at the cytoplasm. So the cytoplasm is a gel-like fluid in which the cell structures are suspended. So this stuff here, all right? So in here, we talked about the nucleus and the rough ER and the smooth ER and the Golgi apparatus and mitochondria and lysosomes, all right, and even the centrioles and stuff like that. Okay, so we already talked about that in lab, so let's skip ahead. If you have my notes, I'm going to skip to different parts here. So next are peroxisomes. So this is showing a peroxisome. So peroxisomes, these are vesicles that contain enzymes that break down poisons. So as the name implies, perox, so we're talking about peroxides and alcohols. So they break down peroxides and alcohols, which can be products of our metabolism. So to avoid them developing in toxic levels, we have these peroxisomes. Those enzymes break down those peroxides and alcohols. Next is the cytoskeleton. So you see these little fibers here and there. That is part of the cytoskeleton. So this is a meshwork of fine fibers in the cytoplasm of the cell. Its main function is to provide support. So if we go to this picture here, you can see that cytoskeleton pretty well there. Now, if you have my notes, I talk about the different kind of filaments. I'm not worried about that. Let's just skip ahead here. I'm going to skip way ahead on a lot of these here and go to appendages. So appendages are structures that stick off of the cell. And the first one I'm going to mention here are cilia. So cilia are short, numerous hair-like appendages. These are going to line our nasal cavity, they line our trachea, so our upper respiratory tract in a lot of ways. And what they're going to do here is they move substances. That's what cilia does. So cilia also lines the fallopian tubes there. It's going to move the egg or embryo. The cilia that's in our respiratory tract here moves mucus. So when we inhale air, we don't just inhale that air, we inhale anything dissolved in air. And so that mucus is there to clean and filter our air. And ciliary action is going to move that mucus to our throat, our pharynx, and then we're going to swallow it. I just swallowed some mucus there. And so that mucus is either drained out of my nasal cavity or up from my trachea to my pharynx, and I just swallowed some more mucus. In fact, we do this throughout the day. We swallow mucus that has been moved by ciliary action from our nasal cavity or our trachea to our pharynx. All right, next is flagella. And flagella are long whip-like appendages, generally few in number. They're going to move a cell through its liquid environment. All right, so this is showing the flagellum right there, singular. And the only cells that humans make that have a flagella are sperm cells.